pointer. Thank you. Good afternoon, everyone. So today I'll be presenting on the bacterial flagellum, and you could see in this video an example of bacteria swimming with the flagellum. And my talk will be investigating how bacteria build these structures, these so-called nanomachines. So here's a brief schematic of what a flagellum looks like. And you can see it's comprised of three parts: the basal body, where the motor is located, the hook, which provides a flexible connector for the rotational energy to the filament, which is the propeller. And the difference between our flagella in our cells and bacterial flagella are they assemble here at the cap. So they assemble very far outside the cell, which presents two problems. One, protein must be exported from the cell. And two, that protein has to go through this narrow channel, which is only about 2 nanometers in diameter. So these subunit proteins must be unfolded as they're exported from the cell. In the past, there were two models for assembly, one called simple diffusion, which, as its name suggests, said that subunits simply diffused through the channel to the tip during assembly, and then pushing from the base, which was a more energy-intensive mechanism in which energy was spent by the cell to push the subunit to the tip for assembly. About 2 years ago, a Harvard lab discovered that flagellum growth is independent, actually, of length, which disputed then these past two models, since if flagellum growth was dependent upon length, both of these mechanisms would slow down as the filament grew longer. And so it forced us to re-examine two fundamental questions. How do subunits move from the base to the tip, and how is that process energized? And my lab proposed that it was due to a chain mechanism, which just says that subunits form a chain in their unfolded state through the channel to the assembly tip, where instead of being exported out of the cell, they are refolded into the structure by this cap. So first, the subunits are unfolded and they're bound to the gate. This means that the ends of the protein are available. And so one subunit in the channel can then use one of its ends to capture the end of the docked subunit, providing a head-to-head linkage. And this forms the chain. The chain then moves up to the assembly point, where it's anchored by refolding. And so then the question became, where is the energy for this mechanism? And that energy actually lies in the thermal movement, the thermal energy of the subunits in the chain as they move through the channel. And so the subunits actually power themselves moving through the chain to the crystallization point. My research focuses on this first step right here, so how the gate recognizes subunits and what that means to assembly. And so we found that there was an amino acid motif, 5 amino acids long, that we called the gate recognition motif. It's highly conserved across protein subunits in this system, as well as across bacterial species. And the structure of this gate, shown here, showed that there was a surface-exposed hydrophobic pocket. And our motif is hydrophobic, so we hypothesized that the motif bound in this pocket. And to confirm that the motif was important, we did a few sets of experiments, the first showing that for a fairly transient temporary relationship, the subunit and the gate have actually a pretty high affinity, and that if you mutated the motif or got rid of it completely, you severely attenuated export. And then this led us to the question, all right, so we know the subunits are binding to the gate, and we know that it's a fairly tight binder. So what effect does it have on the protein dynamics of this gate? And to evaluate that, we used a technique called NMR, which uses magnetic fields to then test chemical and physical properties of a molecule, in our case protein. And here you can see a spectrum of such an NMR experiment. In red is the standard unbound protein, and in blue is the protein bound with subunit. And so what we can get out of this is that when subunit binds to the gate in preparation for export and assembly, there's a major change at the gate. So now we hope to be able to further explain this change so that we can further explain assembly of flagellum, and that way we get a better understanding of bacterial cell biology and protein export. Hopefully in the future we could then use this as a drug target or to study other organisms that use flagellum. Thank you. No, so once the filament assembly begins, they can start rotating. And actually, in an environment when your filament gets really long, so it's about 10 microns in length, which is 20 times the size of an average bacterial cell, it'll experience shearing forces and will start breaking off. But because growth is constant, that's happening as it's rotating. So both processes are happening concurrently.
I have two questions.
Okay.
When you said that those subunits are linked tail, the tip to head, and then they start folding in the extracellular, does misfolding of those peptides occur?
Well, so the cap protein is actually there to prevent exactly that. So the cap, which is what's called a cap foldase, is an enzyme that basically controls folding at the tip so that structures fold correctly or subunits fold correctly into the structure. But otherwise, yes, misfolding would then mess up the structure and probably mess up the chain mechanism as well. And your second question?
The second one was, you mentioned that for the gating protein that there's a high affinity. So do you have any thoughts as to how, since it's such a low KD, how might it be released then?
So we think early analysis of the KD shows that the forces generated by the movement of the chain are actually strong enough to pull subunit from the gate. So the forces generated as it's moved through are an order of magnitude greater than the force that's actually holding it up to the gate. So it's a pretty powerful mechanism.
Might be a silly question. You mentioned that it breaks off sometimes at the end, but it grows constantly. So if the cap breaks off, then you said that it's growing by the cap. Is it reassembled in the middle when it breaks?
So cap proteins, once it hits the filament stage again, cap proteins are constantly exported along with the filament subunit. And when they hit the cap would recognize them as another cap protein and just kick them out of the structure. So it wouldn't allow them to refold, even though in some organisms there may be examples of the cap actually folding into the structure as well. So that's how it would overcome the breaking off and then having to restart the process.
May I ask, speaking as someone who finds it quite difficult to grow hair, what is the advantage of actually growing from the tip, given that you have to do all this, rather than growing from the base? And the old stuff breaks off at the tip or whatever?
Very good question. My guess would be it's how the system evolved, and when it hit this point, it was seen as well enough where there wasn't a selection on something else. With that being said, eukaryotes have decided to do it the other way. So we build our flagellum from the base. As to why specifically this would impart a certain advantage, I can't say.
Thank you.
You're welcome. Do we have time for one more?
Yeah.
Okay.
So you mentioned in passing that you said the energy for the assembly came from the movement of the bottom units through the flagellum. Do you think you could just expand on that?
Yes, so this is pushing my bounds of the understanding of the physics of everything. I just started in October, so bear with me. But as like a self kind of promotion thing, you can always read the paper and find out. My understanding is just like any chain, when you get to a certain point, you get really taut. And that's then, as the subunits would be refolding into the structure, that chain would be pulled tight and then would go back and go slack. And it's in that process, that constant process of going tight, slack, where you have this movement as it moves through. And that's the entropic energy then needed to move the subunits forward. Hopefully I didn't insult physicists, since especially too much for that explanation.
I'm too passe, it's quite safe.
Okay.
Cool.
Right, thank you.